Okay, thank you so much for this very nice introduction. Nice to see you again. And yeah, happy to be back. I wish it was physically. I had the great opportunity to visit Dalton Conley's lab in 2018, but obviously it's a genuine honor to give the Notestein seminar today. And I want to talk about genetics and demography. I'm in Paris at ENSAE, the École nationale de la statistique et de l'administration économique, and some other small units like CREST, Jeunesse, and IP Paris. And I'm working with the research group Social Science Genetics Paris that I'm heading, and I founded 2 years ago. This should be the core group. It's an interdisciplinary and an inter-university group. So Baptiste, for example, is working at the Brain and Spine Institute. Zachary van Winkel is at Sciences Po, and I want to highlight the contributions of our PhD student Tobias Wolfram to all of the new work. He's an excellent sociologist and statistician. And obviously, there's no social science genetics in Europe without Melinda Mills, who is currently the head of the Leverhulme Center for Demographic Research. And I want to thank her and all our funders for the joint work. So there's a little agenda for today. I want to talk shortly about genetics and demography, a bit more general. Then I want to update you on recent findings regarding fertility specifically. First, fertility timing, for example, the age at first birth. That is obviously a big topic for demographers since the dramatic fertility postponement of the second half of the 20th century, but also fertility quantum as completed reproduction. And this, as it has been recently highlighted, that this is also of great importance for population genetics, also in terms that this is an indicator of currently ongoing potential natural selection. And finally, there are some promises, original promises of social science genetics research and in terms of applications. And I want to highlight some of them in the context of social demography. So you might know this slide, but I want to highlight that today and colleagues have already in 1970 in population studies concluded that social scientists are not interested in any aspect of human biology and often deny the relevance of human genetics. However, it has been long recognized that there is a wide range of problems in the human sciences where an understanding of both sociology and human genetics, their concepts, their methods, is necessary. So I just want to say that I don't think this is by chance that this is published in population studies first, because demography strikes me as a discipline that's genuinely in the social sciences very open to new methods and data. And second, if we look what we are interested in, like fertility and mortality, we can intuitively say that these are outcomes that have a genuinely biological, but also behavioral component. So, so here's the data, and this is not all of it, but there's a growing number of sociogenomic data sets out there. With sociogenomic data sets, I mean data sets that don't include genes as biomarkers and whole genome data. And at the same time, a lot of social science information, non-genetic information. This is not only like medical data sources anymore since a while. It's classic sociological and economic data sources such as the Understanding Society, the traditional British household panel study has been retrospectively genotyped. Now also the German PODOR, the GZUP. There is a paper out in the last 10 days, I think. The British birth cohorts is a super homogeneous data set since they followed up all individuals born in a specific month in 1958 until today. Very exciting data set has been genotyped retrospectively. The Lifelines in the Netherlands, Twin Lives in Germany, and of course in the US, the Health and Retirement Study and Health. And it all accumulates in the mega project UK Biobank with more than 500,000 genotyped individuals and thousands of non-genetic variables. So it's not limited to that. It's a bit like Asterix and Obelix. So France are the outliers. There's no genetic data yet, but let's see what the future brings. So the question is obvious, that how can we actually benefit from this? How can we combine insights from genetics? Originally, we were mostly wandering like into social demography, but it becomes also vice versa more interesting recently. So mortality, for example, is, I think, has been for a while already in biodemography been highlighted as something like what's the biological component. And I think this little worm, the planarian worm, plays a major role, which lives apparently forever. And the rumor is that Jimbo Pal might have one in his basement. So what we start to do now is to look whether genetic differences between individuals in a population explain differences in their lifespans. And since the prospective genotyping studies, the good news is that the participants are still alive, most of them. Those have been retrospectively genotyped, and the trick for the genetic discovery is that there's a line now on the slide, but is that in biology, so for example, for height, the best predictor of your height is the average height of your parents. So in these designs, this is retrospectively, the children's genotype is regressed on the parents' lifespan. And studies find that there are obviously some health pathways between genes and longevity. So there is not a specific gene that gives you the age. But there are also behavioral mediators, such as educational attainment. And Dalton Conley wrote a great commentary in PNAS about highlighting both the creativity of this parental design and also the shortcomings, for example, excluding zero parity, so childless individuals. Also, in terms of migration, actually, genetics can be extremely insightful. My intuition is that large parts of migration history, obviously, are rewritten. So the path, where do we come from, but also mating patterns, not only like that the Neanderthal mates with Homo sapiens, but also more less dramatic findings that in the UK we have Roman DNA on the Y chromosome, but not so much Viking DNA, but British X chromosomes are found in Scandinavia and so forth. So large parts of history are confirmed or rewritten these days. And also in sociodemography, there are very interesting contributions. One is Zachary van Winkel from Sciences Po, works on family demography, and constructed actually a measure of life course complexity. So our life's getting actually more complex with the onset and leaving partnerships and birth timing and so forth. And together with Dalton Conley, he has been able to show that genes are actually predictors of this complexity. And also Ledley and Conley and colleagues have done something also very important. We have the finding that skin color is associated with health outcomes. And the question is, what happens if we could control for genes and identify non-genetic influences on that, that would indicate that this is probably also due to stress and discrimination and so forth. So if you want to get into this topic, I mean, you are close to the source. I really recommend everybody, also my students, the Genome Factor is fantastic from Dalton Conley and Jason Fletcher. I heard your Paige Harden visited Princeton not too long ago. Very exciting contribution all over the news, also considering actually the discourse that's happening surrounding these topics. And as mentioned before, I sneaked in a book from us, published in MIT Press, if you want to get hands-on experience to that topic. So I now want to talk a bit more about fertility timing, since the age of first birth is a very important topic. So in general, is fertility in the genes? A question asked by a very famous demographer, Hans-Peter Kohler, in 1999 already. And we know quite a lot about how social and institutional factors influence and shape fertility tempo, like the age of first birth, or fertility quantum, like the number of children. For example, if you stay longer in education, you will probably have your first birth a bit later, and also fewer children, especially women. But surprisingly little is or has been known about biological genetic predictions of fertility outcomes, at least on a population level. So this is very interesting, and it's also important. So even as a social demographer, I would be curious whether the basis of the differences that I observe between people is due to genetic differences or associated at least with genetic differences or not. Also to identify my own genuine non-genetic contribution. Space for contributions. There's obviously a medical story in there. We want to know about infertility and fecundity, actually, also as a function of age. And there's an evolutionary biological story that I wish to highlight a bit more later. So is fertility in the genes is a very interesting formulation. So for example, the number of hands that we have is in the genes, but typically this question is answered with a measure called heritability, and the heritability of the number of hands that we have is 0. The reason for that is that this is an anthropological constant. There is no variation, and heritability is a measure of differences. So it's basically how much of it's a population measure. It's a demographic measure, actually, if you will. It's the differences in a population, the variance in a trait. For example, fertility, how much of that is associated with genetic differences. Interesting for fertility is that it's a fitness-related trait and mortality too. So based on Fisher's fundamental theorem of natural selection, we might expect that such traits become like the number of hands we have and heritability goes to 0. Because over time, variants might be taken out because specific genetic variants are inherited more frequently, and not so beneficial one not. So it has been claimed that this might be a reason why this research has been neglected for a while. Theoretically, it's not expected. However, if we look up at based on a meta-analysis, a fantastic study I find for Poderman of all twin studies ever conducted until 2012, there's a homepage and you can click on the outcome, your favorite outcome, and you get the meta-analysis right away. There have been 280 studies with around 30,000 twin pairs conducted on fertility behavior in general or various fertility outcomes, and it comes to the fair division that around one third of the differences in fertility behavior are associated with genetic differences in a population, 1/3 with environmental differences within the family, and yeah, a bit more than a third, but it adds up to 100% due to between family differences. So genes appear to matter. And what is interesting also in the context of Fisher's theorem is that it appears that genes become more important over time. So here we looked at the UK Biobank and estimated heritability as a proportion of variants that can be related to genetic variants in the age at first birth over time. So on the x-axis, you see birth cohort. On the y-axis, you see SNP heritability. This is not based on twins, but it is based on the whole genome. And heritability almost more than doubles here. So it's definitely a significant increase. So this would mean that the association between genes and age at first birth even got stronger, up to explaining more than 20% of the variants in the age at first birth in the most recent cohorts within the biobank. So this is a strong motivation to conduct genetic discovery studies. I want to highlight that this sounds very firm and maybe mysterious. How can we do this? So we try to establish a link between specific genetic variants and an outcome, in this case, age at first birth, but it's a very, very simple design. It's an association study, so it's a regression model with an intercept with a genetic variant. Most commonly, those are SNPs, single nucleotide polymorphisms, instead of monomorphisms that have no variation, and a vector of control variables, which are supposed to be orthogonal to the genetic variant in terms, so basically gender for the autosomal chromosomes, birth cohorts, but we come back to that, and the principal components which are of the genetic data which are supposed to approximate cultural differences. Importantly, as well is that we talk about millions of genetic variants, so we cannot fit them jointly. So these are really just repeated simple regression models with Bufferoni correction for significant threshold. I'm highlighting this because it's also becoming, it's becoming serious as genetic prediction enters everyday life, but researchers also take it very serious since a while. So this has been highlighted recently also in a fantastic review paper by Young and colleagues. What we pick up in these associations is not only direct effects, causal genetic effects, even if they are mediated, but also indirect effects. For example, our relatives around us, our parents or our siblings, might share the same genes and might act on that, and the behavior might influence us. And there is probably still confounding with environmental factors, correlations with region, with culture, with behavior, with neighborhoods, that is not accounted for by these principal components that are supposed to do that. And there's also assortative mating. So I'm highlighting the limitations before I come to the results. I just think this is very important. And another important part is, this is a fantastic website by Charles Rahal and Melinda Mills, the GWAS Diversity Monitor. And there you can see a breakdown, frequently daily updated, of the demographics of all discovery studies conducted. And the demographics are very selective. So more than 70% of all studies are built basically on 3 data sets. And in general, there are only weird people like Western rich countries. And importantly, these results cannot be generalized. However, keeping that in our mind, we did it, and we conducted another GWAS actually on fertility onset, so the age at first sex and the age at first birth, even though across the past century they have, with the introduction of very efficient contraception, the association between both has diminished a bit. And we find genetic variants associated with both 272 for the age at first sex, which is partly due to the fact that this is based entirely on one data set on the UK Biobank, which is homogeneous and likely triggers more significant findings that, compared to a meta-analysis of 30 studies or more that we did for age at first birth, where heterogeneity across different studies averages effects depending on gene-environment interaction and similar phenomena. And importantly, and we come back to that in the application section, many people are interested in prediction for various reasons. But the question is basically, okay, if you go to a completely independent sample and you don't know anything about the people except of their genotype, can you predict from that, based on that, their age at first birth? And so the polygenic prediction, so you sum up all the effects of all the genes, because there's no single gene, it's thousands of genetic variants important, is up to 5% for age at first birth out of sample and 6% for age at first sex, which is quite substantial. So in the previous GWAS, this was 1.5%, and this might be very important for some applications that I will introduce later. So in this study, we find that these polygenic indices are actually predicted out of samples, and the findings on the SNPs are also, again, like first biological. So they belong to genes that relate to puberty stimulation hormones, to implantation when the embryo meets the uterus, or for infertility and spermatid differentiation. So some findings are also unique to man, and yeah, this is also beyond genetics, obviously. Still also within science, an underestimated fact, infertility is largely seen as a female story in many contexts still, but even on a genetic level, we find that we can show that this is relates to both sexes. But it's not only biological. Genes for fertility timing are also correlated with education, with externalizing behavior, and also with health. health outcomes. Like interestingly, a later age at first birth is correlated with parental longer longevity, potentially indicating some sort of life history trade-off, and with reduced, so it seems to be protective for health. So these are the recent findings on fertility timing, but we were also investigating the number of children ever born and childlessness. So it turns out we had an initial paper from 2015 where we discussed it, but now we basically can think of a GWAS, of a genome-wide association study, as a scan for currently ongoing natural selection. Why is that the case? So in evolutionary biology, fitness is basically mainly determined by mortality, surviving, or by fertility. And in the 20th century, mortality really went down, especially child mortality, mortality beyond the end of the reproductive lifespan. So an idea was that nowadays there should not be so much selection pressure anymore. On the other hand, the question is still, do you have children to transmit your genes? So if there are specific genetic variants that increase the number of children that you have, those will be more systematically, those will be more frequent in the subsequent generations, indicating allele frequency change by natural selection and therefore currently ongoing evolutionary processes. So consequently, we did another GWAS as well, with a massive sample size of 785,000 individuals. We found 270 SNPs associated with a number of children across more than 40 studies. And interestingly, that would indicate that the genetic, let's say, genetically we should have more children nowadays. But as we know, as demographers, in most population, it's the other way around. And the question is, how does that make sense? And it turns out that this disagreement between genes and environment, like the diverging trends of what we observe and what many studies have indicated before in some respect, like for education, for age at menopause and so forth, is possible, at least for two reasons, for many reasons. But the first is that the environment still has a stronger effect. So in Holland, for example, in the last 200 years, the Dutch grew in height 20 centimeters. They were smaller than the US population, and now they are the tallest people on planet Earth. Height, however, is 80% heritable. And the reason is that the variance that is created by the 20% environment is just overriding natural selection or driving it. There's also evidence that natural selection contributed, but for sure not that much. So if we think about educational expansion, economic situation, sexual liberation, introduction of effective contraception, these trends might just, or in combination, these trends might just be stronger. But still, we are lacking the evidence for natural selection on the molecular level here. And the second reason is that it's absolutely possible and rather likely, and I show a study on that later as well, that given the changing environment, it is not only additive, but different regions are under natural selection, or different regions of the genome across different points in time, depending on the environment, and that basically these are only snapshots and there are no long-term trends. So, but what's the story here? Can we say, while we observe decreasing fertility, that on the molecular genetic level this should be increased? And in this study we actually do that. So in two independent cohorts we created these polygenic indices basically as a sum of allele frequencies, since they are multiplied with a constant with the summary statistics of the GWAS. And what we find is that these scores, these indices, increase in the population, in the health and retirement study, and in the UK HLS and Understanding Society, in both cohorts, both for men and women, over time. So genetically, these fertility increasing alleles are more frequent in more recent birth cohorts. indeed. So further highlights of this study are that there is the top hit from the famous same-sex sexual behavior, GWAS, is also one that prevents us from being childless. Then there is a genetic correlation with shorter reproductive lifespan. So later age at menarche and earlier age at menopause. So there seems to be a trade-off between duration and intensity of reproduction. And an absolute highlight, if you ask me, is that so Ian Madison is the lead author here, and he also works with DNA from long with historical DNA, and there's one specific. So that one of the SNPs loads on the fat gene, which is important for saving fat. And this has apparently been under selection for thousands of years now. And the numbers add up. So the allele frequency was 10,000 years ago, 10%, now it's 70%. That fits with the selection gradient that we can estimate and so forth. It's an interesting story because as social scientists, we are very, very excited to get something under selection consistently, because we also can show that there's a lot of heterogeneity between countries, for example, which SNPs are associated with fertility. But the biologists are rather like you should highlight that it's not the same biology over time. So that general question of selection, that was very inspiring to subsume this and combine this with demographic research under a general question of selection issues. Because in genetics, in the beginning, we were very excited to get any genetic data to do our analysis, but especially with the arrival of the UK Biobank, I guess, with more than 500,000 individuals, but also with genotyping, all these representative social science data sources, we got a bit more picky, and the response rate in the biobank is like 5%, and everybody has basically college degree. So there was a special issue in the International Journal on Epidemiology still discussing whether representativity is necessary or should even be avoided, and there are more papers discussing that, an issue that demographers are much more equipped. And as I said before, Melinda Mills and Charles Rahal also highlight really the selectivity of the world population, which is especially a problem or a challenge because these GWAS results cannot be generalized across ancestry group. But the question is, next to these selection questions, is there a genuine selection issue within the data? So we know that there's some sort of natural selection going on, probably, that drives allele frequency changes. And there's also evidence, previous evidence on that regarding genetic predispositions linked to genetic variants linked to educational attainment, that the educational indicating that the educational polygenic score, polygenic index, also decreased over time. And Ben Doming once said, this looks to me like a Rorschach test. Some people see natural selection, I see mortality selection, because people have been genotyped retrospectively. And those older people that survive were probably not smoking and most likely are higher educated as well. So there are many mechanisms that, despite of selection issues, of response rates might lead to selection across birth cohorts, like natural selection, mortality selection, different patterns of assortative mating, migration, or genetic drift, which is basically random changes. So what we did is we did a heritability and a genetic discovery study and the outcome is birth cohort. And we found actually that birth cohort is heritable. So the birth cohort you are born in has specific alleles more frequent than others. And this is a linear model. So we find a heritability of 3%, highly significant without principal components, which sounds maybe low, but if you think about it as pure bias, that might be effective. And even if we include principal components of the genetic data, like the first 100 that are supposed to explain most variant variants, we still explain 2%, 2.3% of the differences in birth cohort by genetic differences. We do not find specific genetic variants significantly associated after Bufferoni correction. But one question was like, if we look at this heritability, can we explain this given our knowledge about genetics of fertility and our knowledge of genetics genetics of mortality? And we can explain around half of the cake, if you will. So, and 50% remains unknown. So this was a bit, for us, a bit surprising, also mathematically, because the genetic discovery studies usually control for birth cohort. And still we have such a strong overlap with findings for specific phenotypes. So in general, we find also genuinely genetic change in, or where the mechanisms are genetic, change in allele frequencies over time. We can partly explain it with demographic measures like fertility and mortality, but 50% remains unexplained. We also looked whether these genetic effects are correlated with other outcomes like height, education, and so forth. And so far, for 22 out of 30 tests, we find a highly significant correlation. So we might have discovered something that is a bias in different genome-wide association studies. And we also find that principal components partly explain heritability. So this is interesting because principal components are usually thought of as tagging geography. Okay, so we wish to provide there a summary statistic that can help with in the genetic structural equation model developed by Andrew Grotzinger and colleagues to control for this additional bias and further study this. Yeah. Okay, thank you very much. And the time is okay. Maybe I'm fast. So finally, I wish to discuss some applications of this research in social demography. So I highlighted already two studies, both involved Dalton Conley, and in general, there were a couple of very clear promises in the beginning of this research. In 2013, the first genome-wide association study on educational attainment has been published in Science, and before there were papers discussing the potential of this research in the integration of genetic information. And these promises were partly methodological. So, for example, thinking about genes as a confounder, we have the initial statement by today that some people just ignore the relevance of genetic factors, and maybe that's not bad. If I'm a social demographer, hey, I might just care about the social, but if I ignore the genes, I would like to actively ignore them. I want no bias. I have to acknowledge in my discussion section that genes exist and that they might bias my results. But if I can integrate them in my regression, they might also just improve my social estimate. Then genes for causal inference. This is, I guess, was one of the big promises, especially from economists, the fantasy that we now have an instrumental variable for basically every outcome, and a bit more towards substantial question is gene-environment interaction. To what extent are environmental treatments sensitive to the genetic differences in a population and vice versa? Jason Fletcher did some amazing work. I found it's the greatest example that, for example, if you change prices for smoking in tobacco, tobacco prices, sorry, this is you think it's a rational choice that people quit smoking, and they actually do. But if they have the genetic, genes that are important for addiction, they don't. they don't quit even though you charge them more. So this is quite important knowledge for smoking prevention, and also for rational ideas about rational choices, I guess. And there are indeed new perspectives, especially in the context of prediction, that I want to quickly discuss as well. So let's first talk about genes as a confounder. And so I think Alice Goises, she's currently in UK, did some fantastic work on a question in demography combined with public health, namely that there's quite a clear association between parental ages and child well-being. So it turns out that children of very young, and with very young I mean teenage parents, and of very old parents, and when I say very old, I mean maybe older than 40, tend to struggle a bit more than the children of their respective peers in terms of socioeconomic status, in terms of health, in terms of mental health. And we also have interdisciplinary explanations for that usually, like young parents have maybe a lower socioeconomic status that might prevent them from nurturing them as competitive, and or they might be still in unstable relationships. And especially for old parents, there's a story of declining reproductive health coming in, also specifically on fathers, because spermoids are reproduced every day, and there might be mutations across the life course, so-called novel mutations that don't affect yourself, but they are inherited. And those might increase your chances, for example, for schizophrenia. So it's quite important to know about this association. If you have a shift in the age at first birth, for example, across 4 decades of 4 to 5 years, because you want to know what the exposure will the next generation have a 5% higher prevalence of schizophrenia or something. So a lab in Australia did the math regarding the mutation story, for example, and they know how many mutations you have approximately across generations, and what the potential effects are. And they said, yeah, the math doesn't add up. There must be something else going on. What about confounding? Because if you scale high on the schizophrenia scale, you might have your children quite early because you have very impulsive behavior, or you might struggle to find a mate. So you might have it rather late, but maybe not just in the average. So we calculated the polygenic index for schizophrenia and plotted it across age groups. And it turns out it has the same U shape as the observed association between the ages. So on the x-axis here, you see the age at first birth in age groups younger than 20, and on the maximum older than 40. And on the y-axis, you see the average polygenic index of the respective age group. And you find basically evidence that mothers with a higher genetic schizophrenia risk have their age at first birth very early or very late, and therefore inherit the genes to their children, who are more likely to be observed to develop these symptoms. So to some extent, at least, genes confound this relationship, and shifts in parental age have a lower effect on child well-being. But in demography in general, we are also very interested in changes of allele frequencies. So we might take a GWAS on birth cohort to control for fertility, for changes in allele frequencies that might be important for biasing trends that we observe, like increasing or decreasing fertility. I just see that I'm a bit late in time. I try to be faster. So the second is gene-environment interaction. And for example, in the discovery of fertility, this is the well-established U-shaped trend in age at first birth. Some people forget that in the first half of the 20th century, age at first birth was quite high as well, by the way. And so we have the U-shaped trend in all the countries, but also on different levels. So the age at first birth, mean age at first birth changes across birth cohorts and countries. So what we did is we were looking whether we explain more variation within the same birth cohort and the same population in age at first birth and number of children than between them. And what we find is that if we pool data from 7 population and all birth cohorts, we can explain 10% of the variation in age at first birth and 5% for number of children. But this doubles within birth cohorts for age at first birth and is 5-fold as high for number of children. So this basically implies that different genes are associated with fertility. We had the same for education, and partly explains why discovery is very hard, that there's no nature versus nurture. It's like the last coffin, and that we have to take these heterogeneities into account when we do cross a birth cohort and cross-country studies. So basically, one fantasy was that we can use genes as instrumental variable, which basically means we have something that predicts education, for example, but not fertility, so at least theoretically. So we can analyze only the fraction of variance in education that's caused by this SNP, and then see whether this fraction of variance still predicts fertility, and that must be a causal effect because the SNP is unrelated to fertility. It's an important question, basically. Let me be a bit fast. I apologize for that, because we have shown in a within twin design that the link between education and age at first birth is much smaller than we expect, and that most of the trend increasing age at first birth in the UK remains unexplained by education. So what we did is we tried this using genes as an instrumental variable based in this study as well. And we find evidence for a bidirectional causality, a small effect of age at first birth on education, but a very, very large effect of education on age at first birth. Like for 1 year more education, you have your age at first birth 2 years later. So that would over-explain basically the trend we've just seen. A potential reason is that the exclusion restriction is not given if we look at indices anymore, because genes are multitaskers and we cannot guarantee that they only influence X and not Y. We tried that in a more sophisticated design where we only take genes from the parents that they don't inherit to their children. So there's no biological connection. Communication must go via the environment, which is actually a very cool finding, a blockbuster once Dalton Conley said. But we run into real weird findings with that as well. We did it on amazing data from Norwegian registers, 11,000 trios, mother, father, child. And we basically tried this technique, and the instrumental variable estimate is right now 7 times higher than of maternal age at birth on children's education is 7 times higher than a standard regression framework. So that is quite implausible and rather calls for, that this is really still to be developed. And finally, in terms of prediction, this is fantastic work by Abdel Abdeloui. So he does some simulations how basically genetic prediction in general might be integrated in dating apps for genetically assortative dating. This is very important for fertility research as well. There's more and more artificial reproduction going on, especially in old ages with the shifting age at first birth. And now we can have information about multiple fertilized embryos, namely via genotyping and prediction. So companies do that. The first child has been born in May 2020. Aurea selected for a low blood pressure and avoiding some cancer risk. However, there are strong problems. I highlighted them earlier, that these associations are just associations. And Dalton Conley again was writing about that in the Washington Post, that we are far behind with some politics. And finally, Robert Plomin wanted to use these predictive scores, or suggest that we might be able to use them for a high school admission as additional information. What we did here is we took 3 measures. First, all polygenic scores that we can think of to predict general cognitive ability at age 11, and we explain around 13%. We can basically predict it on that level. But if we only let the pupils write 250 words in that age, we can predict the general factor in cognitive ability based on machine learning algorithms to more than 60%, almost as high as the teacher's assessment itself. So in summary, genes are associated with demographic outcomes. They change over time and might be partly influence observed trends as well in non-genetic findings. We can use these indices for various applications, and genetics has entered everyday life. Let me close with a quote from Glenn Firebaugh. Apologies for being over time. The best social research most often is research that brings fresh perspective and new insights to old and continuous areas of concern. Thank you very much. I apologize, it's already evening here. Thank you so much for your attention.